All right, part 2 of the blood in physiology, we're going to talk about the leukocytes and platelets. So the leukocytes are going to be, leukocyte is the word for white blood cell. There's a subset of leukocytes called lymphocytes, so don't get those two mixed up. In general, your leukocytes are in the bloodstream, and then they are mobile, so they can leave the bloodstream and infiltrate organs. Some of them will go there normally and take up residence in those organs and just stay as white blood cells in those organs to give immune surveillance to those organs. Some of your white blood cells will just be in the bloodstream primarily, but can migrate into the tissues if they're needed. Elevated leukocyte numbers, right? If your blood shows an elevated amount of any of these leukocytes, that shows that there's some kind of infection, or that you have some kind of problem of inflammation. Inflammation can be caused by autoimmune diseases. It can be caused by allergy. So it just shows that there's some kind of immune system reaction here. It can be elevated in anemia. It can be elevated in leukemia. Leukemia being white blood cell cancer. Decreased leukocyte concentrations can indicate that you have some kind of problem with the spleen, something with the bone marrow or the liver, or you're undergoing radiation therapy, which destroys the actively reproducing cells in the body. One of those actively producing cells in the body would be your hematopoietic stem cells, right? So you would actually be short on all of your blood cells if you were undergoing radiation therapy. All right, so again, here's our composition of blood. We're going to just go through each one of these and talk about their function. So the most numerous in your blood is going to be your neutrophils. The neutrophils are 50 to 80% of your white blood cells. These are very active phagocytes. They're phagocytes. So they're going to take in foreign molecules. They're going to increase in number during infections, and they're very migratory, so they're mobile. And they can migrate into the tissues to fight infection. So they're very good at following the scent of these chemicals, either given off by bacteria or toxins or some kind of foreign invader. They can sniff. They have chemoreceptors that can sort of detect these foreign molecules and follow them. And there's a little video that you can use to see in this module at the end of a neutrophil actually moving around and how it does that. Letter C, the eosinophils. Eosinophils. So eosinophils are not that plentiful, 1 to 4% of your white blood cells. They can be phagocytic, but they're usually not. They are predominantly involved in fighting a parasitic infection. So parasites are usually eukaryotic. So these are eukaryotic. So things like fungal infections or some kind of worm. Okay, so usually parasitic, eukaryotic infections. And what they do is that they take these little granules that are in their cells and they release these toxic molecules. So they release toxic chemicals and they irritate and they try to attack those parasites with these chemicals. So eosinophils, if you see an elevated eosinophil count, that could tell the clinician that you're having some kind of parasitic infection. Basophils are also very few, also 1 to 4%, so not many of those. They are not phagocytic. Oh, I'm sorry, not 1 to 4, 1%. 1%. They're not phagocytic, and what they do is they are also kind of like eosinophils. They can attack parasites by releasing those toxic substances, so they can attack parasites. But they have a role in allergy. They're actually the cells that are contributing to, in the most part, to your allergic reactions. And so why do they do that? they release a couple chemicals that are really important mediators of allergy, which is histamine and heparin. So histamine is a powerful vasodilator. Okay, so it's going to open up your blood vessels. So if you have an allergic reaction, if you're just having hay fever, if you think about what's happening in your nose and in your throat, it gets red and swollen and hot and itchy. That's all due to histamine. So histamine is a vasodilator, so it's going to open up your blood vessels. That can cause edema or swelling. That can cause the redness. That can cause the heat in all of that because your blood vessels are opening up. And then it also can cause runny nose, right? That extra fluid, runny nose, watery eyes, all miserable symptoms. And so that's due to histamine. Histamine also causes itchiness, which is not in the outline, but it does cause itchiness. And anyways, the over-the-counter antihistamines are basically counteracting the histamine that your body's producing. Histamines can make you drowsy by antagonizing the histamine receptors in the hypothalamus. So sometimes if you feel you have allergic reactions, but you're also feeling groggy and tired, that's the histamine. Heparin, the role is unclear, but it's an anticoagulant. Okay, so both histamine and heparin are released in allergy. For monocytes, this is going to be about 5%. And monocytes are very phagocytic. So monocytes are found in the bloodstream. And what monocytes do is blood, and let me just put the word phagocyte. So they will migrate into the tissues. When they go into the tissues, they can either become fixed, they become what's called a macrophage. And they can either be a fixed macrophage, the abbreviation for macrophage is like that, or they can become a wandering macrophage. So fixed macrophages are just there forever. So they are in the organ in one place and they just sit there. So one fixed macrophage you've probably learned is your Kupffer cells that are in the sinusoids of the liver. For example, Kupffer cell. Or there's those phagocytes that are in the lung, in the alveoli of the lung. So those are fixed. Wandering just means that they still have the ability to move around, that they still can migrate through the tissues. So they're not in the bloodstream anymore, they're in your tissues, but they can still wander around, just looking for any foreign invaders. There's a special kind of monocyte called a dendritic cell, or a macrophage, dendritic cell. And the dendritic cell is going to be found in places that are between the outside and the inside of your body. So this is going to be the skin and also in your mucous membrane. Okay, and in the skin, you've learned the name of one of these dendritic cells, Langerhans. Okay, so I'm going to flip to here just to show you that they call dendritic cells because the word dendrite comes from those neurons, right? So remember, dendrites are these short, highly branched processes off the body of a neuron. Well, dendritic cells also have those short, many short processes that come off of their entire body. So they're kind of stretched out and they have their extensions stretched out in all different directions because they're reaching as far as they can away from the main body of their cells to detect anything foreign. So here's our immature monocyte maturing into a dendritic cell. And you can see this cartoon shows you that there's different growth factors that are required for this, or cytokines are required for this differentiation. You don't need to know that though. All right, moving on to lymphocytes. So before we get there, so we can see the monocyte to macrophage. The lymphocytes are two kinds, right? There's T cells and there's B cells. The lymphocytes is the next really big one. So if you look at your total white blood cell count, The most numerous is your neutrophil. The second is your lymphocytes, your B and T cells. So your B and T cells are actually most abundant in your organs. They're going to be found in your lymphatic organs, which is your lymph nodes, your spleen, your tonsils, things called Peyer's patches in your intestines. So they're going to be found in organs they're going to be highly densely sort of collected, these T cells and B cells. Okay, so in your interstitial fluid, this is where you find, so the kind of white blood cells that are in your interstitial fluid would be B cells and T cells. So let's take a look at our next picture here. And these are the lymphocytes. So one thing I want to make sure that you guys understand is lymphocytes are considered specific immunity. And we're going to talk about immunity in the last part of this, so I'm not going to go too much in detail, but specific immunity means that they're not going to react to anything foreign. They're going to be primed to just recognize one particular foreign substance. So a particular B cell, for example, will recognize maybe Staph aureus, but not recognize Staph epididymis. So they're specific as to what they react to, but the nice thing about specific immunity is that they can form memory. So they can remember what your body has seen before and create memory cells so that the second time your body sees that same antigen, the same foreign molecule, you will have the memory cells to elicit a very rapid and intense immune response, a lot more rapid and a lot more intense than you would the first time around, so that the second time around you don't actually feel sick at all because your immune system can clear the infection immediately, or very, very fast. So the two different ways that these specific cells work, they're very, very different. So the B cells are going to, when they contact their antigen, so if you follow the outline, they're going to contact the antigen. And just let me make sure you guys know what antigen is. So an antigen is going to be a molecule that is going to be a molecule that the immune system can recognize. So antigens can be foreign antigens. They can be something on the surface of a bacteria or a virus, or they can be self-antigens, which means your own cells. They can recognize your own proteins, your own molecules. So your immune system knows to leave your own molecules alone, to leave your cells alone, but to attack something that doesn't look right, something that's foreign. So in general, we use the terms self and non-self. Non-self is something we attack. Self is what we leave alone. So B cells are able to detect these antigens, and then they differentiate. So when this B cell detects something, it differentiates into a plasma cell. The plasma cell is the active form of the B cell, and it will start to create antibodies. And you can see the antibodies have this Y shape to them. So what antibodies do is they tag the invader. So if we have a bacterial cell here, the antibodies will attach to that bacterial cell. It'll coat the bacteria in antibodies. And then what that does, antibodies do several things that you'll cover in microbiology if you have to take microbiology. But what that does is it signals to, let's say, your neutrophil. Okay, let's say we have a neutrophil here with a many-lobed nucleus. This is your neutrophil. It'll say, the neutrophil will go, that thing has antibodies all over it. Let's phagocytose it, right? So it'll eat it. Macrophages do the same thing. The phagocytes of your body will recognize something with those antibodies on it and take it in. So antibodies act as a tag, right? So whatever's tagged, your phagocytes will eat, and you'll get rid of those invading pathogens. So the B cells don't kill, they just secrete these antibodies, and the antibodies tag, and then your phagocytes will destroy. So it's the neutrophils and the macrophages and the monocytes that actually destroy. Your B cells just produce the antibodies that tag. T cells are different. So T cells, when they get stimulated by an antigen, they differentiate into different kinds of T cells. And I'm just going to talk about 3. So one is called the cytotoxic T cell, or the CD8 positive. So the CD is a receptor type on its surface. So this one has CD8. And then this one has a different kind of receptor, right? And this will be CD4. So they're differentiated in this way because those receptors have different jobs. So the cytotoxic T cell is going to kill. So we can see by its name, cyto is cell, toxic is toxic to cells, it kills cells. The cells that it kills is going to be your cells. So these T cells can recognize self and non-self. The T cells, the main job of T cells is to take care of your cells, the ones that are sick, the ones that are infected, the ones that there's a problem with your own cells. Your T cells can recognize that your own cells are infected or they're cancerous and kill them. So these are going to kill cells, and they're very active against virally infected cells because viruses actually go inside your cells. And then they hide inside your cells and they reproduce inside your cells. So your cells will actually look a little different. They're going to have different proteins on the surface when they're virally infected. And so your CD8 T cells can detect that and destroy that. So they're very, very good for viruses. They're also very, very good for precancer or cancer, because when you're developing cancer, when your cells are being transformed from normal to cancer, your T cells can recognize that change. So these are very, very good for viral infected cells, cancer cells. And then also, FYI, foreign tissue. So if you get a transplant, your CD8 cells can recognize the difference between that transplanted tissue and the normal tissue, and then try and reject the transplanted tissue. So that's not good but it recognizes that foreign tissue. CD4 is known as a helper. So this is going to help activate the entire immune response. So it activates the CD8 cells, it activates your B cells, it activates all the neutrophils, macrophages. So it just sends out these cytokines that are going to activate all the other kinds of cells. Then lastly, there is something called a T suppressor, which is not on here. So the T suppressor cells are going to dampen down the immune response. So your immune response is going to activate and activate and activate and be active, but then what signal tells it to all quiet down after the infection's gone? It's T suppressor. So that's what the job of here is. And then there's another kind of T cell called a null cell or natural killer cell. And natural killer cells are the sort of specialized for virally infected. So this natural killer, NK, is a kind of CD8. So it's going to attack virally infected cells, very fast acting. So natural killer. All right, so that is our lymphocytes in a nutshell. And we're moving on to our last topic, which is platelets and hemostasis. So hemostasis is to stop bleeding. And it's done 3 ways. So the first thing you're going to do is the vessel itself will spasm, right? So the smooth muscle here is going to contract to help stop the blood flow going through that vessel. Then our second step is our platelets will come in and form something called a platelet plug. Then our third step is to coagulate. So the plug and coagulation are separate processes. So we're going to take a look at the platelet plug first. So here is our step of platelet adhesion. So let's start really quick with the platelets themselves are fragments from a megakaryocyte. The megakaryocyte was in the bone marrow. There's no nucleus, It has some organelles, it has granules, it has secretory vesicles filled with clotting factors. So one of these is von Willebrand factor. Von Willebrand factor is secreted by megakaryocytes. It's also secreted by platelets and the cells of the blood vessels themselves. And they're going to, the von Willebrand factor is in the blood at all times, but it's going to accumulate at the site of damage because it's going to be attracted to collagen. So, which is here. So let me just draw you an image, right? So you have a blood vessel. Remember that inner lining of that blood vessel are endothelial cells, right? So let's draw some flat endothelial cells just here. Maybe we'll draw, here's the nucleus. Here's 3 of them. And we know that underneath our endothelial cells, we're going to have connective tissue. And one of the major fibers in this connective tissue is collagen. So we're going to have collagen right underneath our endothelial cells. And in a normal blood vessel where it's not broken, the platelets, right, these little platelets are never going to see that collagen. The von Willebrand factor is never going to see that collagen. Only when you have a break, right? If there's a break, a rupture in the endothelium, then the collagen fibers are exposed, and then that's what's going to recruit your von Willebrand factor to those fibers. That von Willebrand factor is going to anchor your platelets. So your platelets are also attracted to the collagen, but they're going to need that von Willebrand factor to stick better. And so there is a problem there actually is a von Willebrand defect. You can have a problem with not making enough von Willebrand factor. It's called von Willebrand's disease. And you have a decreased ability to make these platelet plugs. Okay, so it's really important to maintain the platelet plug. So the first thing we're going to do is make a platelet plug. So again, here's our little picture. Here's a normal blood vessel. We can see that we have our endothelial cells, we have our collagen fibers underneath the endothelium. And just to mention that the endothelial cells actively inhibit a plug by 2 chemicals that they're always pumping out. So normal blood vessels will always pump out prostacyclin and nitric oxide. And those 2 are going to inhibit your platelet plug, your platelet aggregation. So in the situation where you do have a cut and those collagen fibers are exposed, you're going to have the von Willebrand factor active. You're going to have the platelets coming in. And the first step, if you look at in the platelet plug steps, the first step is adhesion. Adhesion just means, hey, I'm going to have some platelets sticking to the collagen. And then there is a second event called aggregation. And the second event, it requires a positive feedback loop. So the positive feedback loop is a loop from thromboxane A2 and ADP. So the platelets will actually secrete ADP. So you see the arrow coming out of ADP. And then ADP is going to stimulate more platelet aggregation. So what aggregation really is just platelets sticking to each other. The platelets will also secrete thromboxane. That thromboxane was going to positively create more ADP. More ADP. More ADP means more platelet aggregation. So you can see there's a positive feedback loop. The thromboxane causes more ADP. More ADP means more aggregation. The more aggregation you have, that means more platelets. More platelets means that they're going to be secreting more thromboxane. So it's kind of like this positive feedback loop to get you a nice, large aggregation of platelets. And that's the plug. Okay, so we have a plug. The plug is not strong enough because we can imagine that the blood is still whipping by, right? We still have blood traveling through this vessel. So these platelet plug can be broken and carried away in the bloodstream. So the clot needs to be reinforced by a second process called coagulation. So creating a blood clot is the synonymous with coagulation. So in order for that process to happen, it's a little bit more complicated, and there's lots of factors involved in controlling coagulation. there are two pathways, and looking at your outline really quick, just understanding what a clot is. A clot is where the blood is going to become a solidified substance called a thrombus. So a thrombus is a solid gel, and that gel is going to contain proteins, fibrin, and it's going to contain cells that are trapped in that fibrin web, and it's going to occur around that platelet plug, excuse me. And so the dominant, so when we talk about a clot, we're really talking about a fibrin clot. So fibrin is the most important, if you can see it here at the end of our pathway, it's the most important protein involved in that clot. So there are this cascade of events to try and activate fibrin. And you might think, so let me just show you the other picture too. So there's two pathways. There's something called the extrinsic pathway and the intrinsic pathway. You can see how the two pathways converge onto the end product of fibrin. So fibrin is so important, and we need to activate it. So you need to understand that fibrin is not going to be in the bloodstream in its active form, because you don't want fibrin to form a clot willy-nilly, right? You need to control when and where that fibrin forms a clot. So we're going to have an inactive form of fibrin in our blood called fibrinogen. Inactive. So we can see here, this is the bigger picture. So fibrinogen is inactive. This is in the blood at all times, because you never know. We never know when we're going to have a cut or where we're going to have a cut. So we have to have fibrinogen everywhere, and only activate it when we have a cut. So there are mechanisms to control this. So what I want you guys to do is I'm going to go through this. You're just going to be able to, you're going to want to know the events here. So where the extrinsic pathway and the intrinsic pathway converge, they converge onto this factor 10. So these are all Roman numerals. So X is 10. I want you to know everything from here out. So know the steps here. And it's just 3 or 4 steps, so not too much. But I want you to also understand just the different pathways. So what we have here are two pathways that are in the bloodstream, and the extrinsic pathway, if you look at the words under coagulation cascade, the intrinsic pathway is where your coagulation factors and chemicals are in the blood at all time. And this is kind of like the workhorse, and it does most of your clotting. Then the extrinsic pathway is going to be activated when there's damage. Okay, so they're both activated when there's damage, okay, but the extrinsic pathway happens very quickly, and it takes a little bit of time for that intrinsic pathway to happen, because you can see it's a little bit longer. Okay, so two pathways. One is very quick when there's damage, the extrinsic, and one is longer, but it's actually the more robust pathway. So they're both going to converge onto factor 10, and I just want you to understand that these clotting factors, and your book calls them coagulating factors or coagulation factors, but you've also out there in the world they're called clotting factors. So they are inactive in the blood. You can see inactive, inactive, inactive. They're always around in your bloodstream, but they get activated when this cascade begins. So when they're activated, you put a little A next to their number. And so you can see that this chain of events, you have one clotting factor activating another clotting factor, right? So for example, number 12 activates 11. 11 becomes activated. The 11 activates 9. 9 gets activated. So it's a chain of events like that. Don't need to worry about the details there. Just know that the clotting factors are inactive in the bloodstream. They get activated by other clotting factors. So they converge on the activation of 10. So 10 is an important clotting factor. So inactive 10 gets created to active 10. Okay, So why is 10 important? Because 10 is what activates one of the main clotting proteins, thrombin. So again, thrombin is in the bloodstream in its inactive form called prothrombin. Prothrombin gets activated by our 10 into thrombin. Why is thrombin so important? Is because thrombin is the last step before it activates the fibrinogen to fibrin. Right? So fibrin, so thrombin is also, the active form thrombin is going to activate fibrinogen to fibrin. Notice that thrombin has the number 2, so technically thrombin is coagulation factor 2 or clotting factor 2, and technically fibrin is actually 1. So it kind of makes sense that 2 activates 1. So that's what we're doing here. So we're having our 2 pathways converge on 10. 10 is going to stimulate our activation of thrombin. Thrombin then goes ahead and activates our fibrin. So those are the 3 major steps that you want to know in this convergence of the extrinsic and intrinsic pathway. So then when fibrin is activated, fibrin starts to build into, on top of one another. So if you watch the Khan Academy video, he does a very good job going through. But what's happening is that you have fibrin in the bloodstream, but fibrin has a little cap on it. So that needs to be removed, and that's how it gets activated. So fibrinogen looks like it has a little cap on it. When that's activated to fibrin, the fibrin cap, the little cap comes off, and then fibrin is allowed to make a fiber. So it lines up and makes a fiber, like a long string, and then that is going to form a meshwork that lies on top of our platelet plug. And so you have these strings, right? But the strings are going to get covalently bound together. They're going to get hooked up together like this by another factor, which is 13. Okay, so 13 is going to come in, it's going to make those covalent bonds, and it's going to make that mesh stabilized and more stronger, right? So it can form that clot. So if you look in your outline, there is a mistake. And so I have under coagulation cascade where I actually had factor 8, but it should be factor 13. So factor 13 is also called the fibrin stabilizing factor, right? So this guy here is also called fibrin stabilizing factor. All right, so that's how it works. And this is our picture of a fibrin clot. So we have this web network of fibrin fibers, and it traps red blood cells, it traps white blood cells, and so a thrombus, which is the technical word for clot, is going to contain a population of just the cells that are around, all woven together with fibrin. So this, coming back to this picture, this is just kind of starting at factor 10. Right, so you want to know this picture. So this is our intrinsic pathway converging with our extrinsic pathway. We're activating 10. 10 is going to activate our prothrombin to thrombin. Thrombin is going to activate fibrinogen to fibrin. 13 comes in and stabilizes our meshwork. So there's a positive feedback mechanism. There's a positive feedback mechanism to thrombin. So thrombin can actually activate more factor 10. So thrombin can activate more factor 10. So we can get a more profound effect on making a clot. Okay, so you can also see that thrombin has another positive feedback loop to our intrinsic pathway. So we have a positive feedback mechanism within clot formation by thrombin. Moving to our, so this formation of blood clot, the clotting factors we talked about, all those numbers are produced by the liver. And as I mentioned before, they're secreted in the blood in an inactive form, and they're activated during the cascade. Again, plasma without our clotting factors is known as serum. And the disorder called hemophilia is where you are born, it's a genetic disorder, in a deficiency in one of those clotting factors, usually factor 8, but it can be, there's 3 kinds of hemophilia. Each one has a different clotting factor that you're missing. And hemophilia is basically where you can't clot properly. Okay, so it's a disorder of clot formation. Hemophilia, von Willebrand's disease, and then vitamin K deficiencies are all known to cause problems in clotting. This is genetic. You're missing a clotting coagulation factor. Von Willebrand's, I talked about earlier, it's decreased. You have reduced levels in the bloodstream, and you don't have the ability to form that initial platelet plug. And then vitamin K is required to make those clotting factors. So if you're low on vitamin K, then you're going to be low on those clotting factors. So those are your sort of diseases of clotting factor. This is a picture of hemophilia. So it can show up as a bruise because we know that bruises are just bleeding inside the skin, right? So we have bleeding under the skin. And hemophilia, it's, you know, if you bump yourself, you can bruise, and then that bruise you'll stop bleeding, and then the bruise will eventually go away. But for people with hemophilia, that you can't really stop that bleeding. It's not more bleeding than normal, but it's just that you cannot clot it. So you're going to have a continuous bruise. And that extra blood, if it happens within joints, it can cause swelling in the joints. So if it happens internally, sometimes you don't even know if you're bleeding internally because it's not something that's obvious like a bruise or a swollen joint. You can just have blood sort of pooling up inside your organs or in your cavities, and that can be very dangerous. Okay, how about dissolving a clot? So I actually don't have a picture of dissolving a clot, but from your outline, there's a molecule called plasminogen. Same thing, it's inactive, but it can be activated to plasmin. Plasmin will dissolve the clot, right? So if we just go back to our picture of a clot, plasmin is going to dissolve the clot when you don't need it anymore. Along with just that normal degradation of the clot, you also have factors limiting clot formation. So just why don't you throw clots willy-nilly? Because your tissues will actively make sure that you don't, to prevent this fibrin from clotting. And one of them is called tissue factor pathway inhibitor. And this is secreted by your healthy endothelial cells, and it inhibits the extrinsic pathway. So I can go ahead and put that on here. So the extrinsic pathway, this is inhibited by our tissue factor pathway inhibitor. And then there's thrombomodulin. So thrombomodulin is also something that's secreted by healthy endothelial cells. It binds to thrombin, and it inhibits both pathways. So you have another molecule, thrombomodulin, right? And that's a good name because it modulates thrombin. So that's going to, so you know how thrombin can positively feed back to your intrinsic pathway, that can stop that. And there's another step for extrinsic pathway. It's not shown, but it can just stop that. It inhibits both pathways for its own activation. So that's good. Let's go into our last little idea here, and that's aspirin. So some of you may know that aspirin is given as a blood thinner. If you have problems with clots, you're given aspirin as therapy. So it just so happens that at low doses, it happens to be an anticoagulant. And the reason that it works is that it's going to inhibit an enzyme called COX-1. And the COX-1 is going to be responsible for making, this pathway here. You don't need to memorize the pathway, don't worry about that, but know that this pathway will ultimately lead to platelet aggregation, right? Vasoconstriction. And those are both bad things if you're prone to throwing clots. So this aspirin is going to stop, so it's negative, it's going to stop your platelet aggregation, and it's going to stop that vasoconstriction. So aspirin does that. So this is the good, it's going to be, low doses are good. High doses, it's weird, but high doses are going to inhibit another enzyme, and it's going to actually increase your clot formation. So if you look at the second pathway here, The second pathway inhibits the aggregation, but since your aspirin shuts this down, it doesn't inhibit anymore. It's going to stop the inhibition, which lead to its free-for-all. Go ahead and aggregate your platelets. So it's a funny little thing here that more is not better, right? You want to take aspirin at low doses because low doses inhibits the enzyme that would induce platelet aggregation. And high doses would do the opposite. High doses would increase clot formation because it inhibits a separate enzyme. All right, I think that's the end. Some review questions. Oops, I'm going backwards. All right. The most prevalent of the leukocytes are neutrophils. What's the primary role of neutrophils? And the answer is letter B. Which leukocyte contributes to the allergic reactions by releasing chemicals such as histamine and heparin? The answer is letter D, basophils. Which type of cell is not capable of phagocytosis? This is letter C, cytotoxic T cells. What converts the plasma protein fibrinogen to its active form fibrin for blood clots? And the answer is letter B. What are the steps of blood clot formation in order? And the answer is letter D as in dog, platelet adhesion, and then aggregation, and then fibrin clot formation. Which chemicals released by healthy endothelial cells prevent formation of a fibrin clot? And this is letter B. Which chemicals released by healthy endothelial cells prevent formation of a platelet plug? And this one is letter D as in dog, nitric oxide and prostacyclin. So one thing to notice here is that you have chemicals that inhibit a fibrin clot, and you also have chemicals that inhibit a platelet plug, right? They're different. So you have inhibition of the plug and also inhibition of the clot. And that is it.